Welcome to you to the 2024 edition of the VG Kulkarni Memorial Lecture, and this is the 23rd edition of our flagship annual lectures dedicated to the memory of the visionary VG Kulkarni, our founder. And over the years, of course, we've had an excellent series of talks covering various important issues in science, technology, education, policy, society, a lot of different topics. And every year it's an honor to welcome all of you to this event. It's always a special occasion. This year it's even more special. It's our golden jubilee, 50 years of our commitment to education, to equity, to excellence. And the seed planted by VGK is 50 years young, I would say. And a special welcome to all the teachers from the MSFDA workshop. This is also the concluding lecture of a teacher development program that we've been running. A new thing this year is a partnership with the Maharashtra State Faculty Development Authority. So we have undergraduate science teachers across Maharashtra over here as well. And this is the last in their series. They've been here for a week. Now, I know that there's going to be a vote of thanks at the end. However, I do want to make a special acknowledgement at the beginning that the fact that we are here is quite remarkable. And as some of you are surely aware, Wednesday night we had an unprecedented spell of rain that literally flooded everything on campus, and the floodwaters entered this place as well. And on Thursday morning when I had come, chapak karke, I was wondering what is plan B going to be. And I must say that our technical services and the cosmetic maintenance team, with all these big fans and heaters and suction things and shampoos and whatever else, have worked some wonders and got this auditorium into a state that we can have the lecture today. So hats off to all of you. A huge shout out to you. Thank you very much. So let's get on with the VGK Memorial Lecture. It's my pleasant duty to say a few words about our founder. He joined TIFR in 1953 and was a very prolific researcher in nuclear physics, in solid state physics, etc., till he decided that his calling was to be in plunging in to the field of science and mathematics education, focusing especially on children and especially on the underserved segments of society. And with that, they set up, Udgaonkar, VGK, others, they set up a small unit that looked at science education issues, in particular the role of language in learning sciences, Marathi, looking at kids across various communities. And, over the years, starting from that, it is those ideas which have given us direction over the last 5 decades to the work we do today. And, no better choice to go back to TIFR. We have Professor Vidita Vaidya here to speak to us today. And before I let Vidita take the stage, thank you so much for coming. But I also need to embarrass you properly. So let me, you've agreed to do this, but it's my chance to also. So I think we will invite the youngest student in HBCSE. Shalu, is that you? I think it's you. That's what established mode told me, at least, that it was on records of the youngest student. So may I request you to please come and felicitate Vidita. But where is this tiny bag that we have? Please. Yeah, come on. Okay, you can come. Yeah, because you're going to be up anyway. No? Come. That's a little token of our appreciation. All these things they are mostly HBCSE products, actually. You can show people what we have. We have a book from the design lab, HBCSE mug, a picture of plants on campus with the location on the map of everything and where the plants are and things like this. So it's all HBCSE magnets which have HBCSE birds on them. Everything's HBCSE products here. This is not. And I also see Anita ma'am here, and that's VGK's daughter. Thank you. You come every year. It's a great support to have you, here for this talk and support us through the year. And I need to embarrass you as well. And, for all these years, I have seen him only for 3 years, but for every lecture in this room, for everything, if you have to bring flowers, if you have to arrange the decoration, if you have to arrange something, who does it? Gajanan. So I'm going to request Gajanan to kindly present Anita ma'am with bouquet and Okay, on that note, let's go ahead, and I'm going to hand over proceedings to Deepa. You can come and formally introduce the speaker. And, you want to sit down?
Stay here.
You can stay here, yeah. You're not taking it long. She's going to do it.
I'm fine.
Yeah.
All right. Thank you, Professor Bhattacharya. And it is an honor to introduce our VGK memorial speaker today, Professor Vidita Vaidya. She is currently a senior professor and chairperson of the Department of Biological Sciences in TIFR, Mumbai. Professor Vaidya received her undergraduate training in life sciences and biochemistry at St. Xavier's College. She obtained her doctoral degree in neuroscience at Yale University with late Professor Ronald Newman. She then contributed her research as a postdoctoral fellow at Karolinska Institute in Sweden and also at Oxford University, UK. In 2000, she returned to India and joined TIFR as a faculty member. Professor Vidita Vaidya is a fellow of 3 Indian science academies. She received the National Bioscientist Award in 2012, the Shanti Swarup Bhatnagar Award in Medical Sciences in 2015, and also the Infosys Prize for Life Sciences in 2022. She was also awarded the Nature Award for Excellence in Mentorship in India in 2019. Vidita's research group is interested in understanding the neurocircuitry of emotion and its modulation by life experiences and the alteration in emotional neurocircuitry that underlie the complex psychiatric disorders, particularly something like depression. So I'm sure we are going to hear a lot about it in today's talk. And Professor Vaidya is also committed to enhancing equity, diversity, and inclusion in academia through practice and professional experiences. So without any further delay, I now request our distinguished speaker, Professor Vidita Vaidya, to deliver the 23rd VGK Memorial Talk. Thank you.
Thank you. It's on? Yeah? If it's too loud, tell me. Is it okay? Okay, I don't want to be too loud. Thank you for this invitation, and it's an honor to be following in an incredible list of speakers that have given this talk over the last 23 years now. I'm in the 23rd in this line, so that's pretty remarkable. I'd like to convey my regards to Anita Patil, who I had the pleasure of sharing a cup of chai with right before this particular talk, and she's the daughter of a remarkable father. I also was reading up about him because I was wondering what I could do in terms of connecting to the talk that I plan to give today. And in the introduction that I read about him on the HBCSE website, something that struck my eye, which was that one of his sources of focus was to actually look at disadvantaged sections of society with a view to looking at what it would require to enhance STEM education to a sector of society that perhaps does not have the access and opportunities that many others take so easily and for granted. So I want to start with this. I was thinking to myself, why would a neurobiologist have anything of importance or relevance to say to educators, educationists, etc. Of course, the easy, simple way would be to say, I tell you about what the brain does as far as learning and memory and acquisition of information is concerned, but perhaps there's something more and deeper that neuroscience can provide as insights. And the reason why I thought I would start with this quote is that science education really doesn't happen in a vacuum. When you're trying to convey something about the STEM disciplines to a world out there, the life histories of the individuals that you're conveying this to matter, and they matter immensely. So this is a popular cartoon. It has been used multiple times. There are issues with this cartoon, but I think it conveys the point reasonably decently, which is our understanding of what equity means, what we perceive as equality in contrast, and what reality may actually be, which is that you might be serving largely this pool, which is carrying a remarkable set of centuries of privilege, and actually there might even be greater consequences that push back and further steepen inequity in our society. And maybe it's something well worth thinking about for STEM educationists as to whether we are contributing to steepening this differential rather than actually reducing the steepness of this differential. Okay. So why should India care about this, and why does it even matter for the average person in India / policymakers / scientists / educationists to worry about this? We are going to provide the world's workforce. This is very clear. This is a 2020 demographic. Of course, the tragedy is this is the excess male population, and you can see that the female population, this is just the excess difference that tells you what we are dealing with in terms of another intersection here of population, which is that the female numbers are slimmer and shorter for a particular reason in our societies and in our culture. But this is the numbers that we are talking about entering the workforce. Do we have jobs for all these people? Given that STEM education is probably a big driver of job opportunities, are we going to be able to provide any potential possibilities for this large workforce that's entering in? Just to give you a sense of a comparison between China and India, this is the sort of numbers you're looking at. So China, as a consequence of when it constrained and controlled population control versus when we have done it, we'll also see a shrinking of that pyramid structure. It will change its shape to being a slightly different shape, but the shape is quite drastically different even from China. And that gives you a sense of what we're talking about. It's not just India's problem, it's actually a world problem, because this is where a large proportion of most other economies have shrinking numbers with a ulta pyramid, in that you will end up with a very large number at the top and less number of people to support this aging population that is going to exist across the world. All right. So why should we think about early adversity and why this might matter? So early adversity has a major role in determining health outcomes across the lifespan. This has been appreciated perhaps most initially by epidemiologists, psychiatrists, psychologists, but eventually also by those who are interested in learning outcomes and educational outcomes, because this is a big factor in determining learning outcomes for the population that enters into the STEM classroom, either childhood or undergraduate or at any stage. So this is a major factor. So this old study, and this is now from a while ago, let me see if I can switch over to this. So this old study from Felaty et al. Is actually based on, so unfortunately a large proportion of the data I'm going to show you is going to be lopsided, in that it's going to come from the West, which also points to a missing piece of information we have in our own country, in that we don't have sometimes the depth of information that we require. So this is a large study that was carried out in a managed healthcare in the US, where they looked at 17,000 people, and it was a survey-based approach where they just asked for questionnaires to look at the number of adverse childhood experiences. And adverse childhood experiences could be defined as abuse, neglect, household dysfunction of different kinds. And so you say if the individual has had no experience of an adverse childhood experience, you define it as a score of 0. And then as they increase, you increase the number to say 2 adverse childhood experiences, 3, 4, 5, 5 plus, etc. With the view of then asking whether there is a correlation between the number of adverse childhood experiences and the eventual health outcomes. The same has been done in a variety of situations, including asking as far as whether this is a factor for early mortality, as well as a factor for learning outcomes. And across the board, this is a common risk factor for almost all psychiatric disorders, from depression to anxiety to substance abuse to obsessive compulsive disorder to schizophrenia, including those that have a higher genetic predisposition. You overlay genetic predisposition with early adversity, and it manifests perhaps in an accelerated manner and sometimes in a worse manner. So it both exacerbates, worsens, as well as sometimes can precipitate psychiatric disorders, including substance abuse. But this also is a major factor in learning outcomes and cognitive capabilities in children, because this becomes a serious risk factor for that as well. So this has been appreciated for a long time, and what is known, and this is of course biased in that it is really looking at psychiatric outcomes in a sense, but as ACE scores increase, you can see that substance abuse, for example, nicotine addiction clearly goes up as there is an increase in the number of ACE scores, alcoholism as well, IV drugs, cardiovascular dysfunction shows up with a higher rate, obviously suicidality and suicidal ideation, but in addition to this, anxiety, depression, etc., all follow this sort of a pattern. This is also true for cognitive function and learning outcomes, with ACE indicators being a major risk factor to determining cognitive outcomes as well. Okay, so what are we talking about? What population is then potentially has a history of adverse childhood experience, and what are the most common adverse childhood experiences? The most extreme ones tend to get the most attention, sexual and physical abuse and trauma, but the most common one is actually neglect. Neglect is the single largest, most prevalent form of early adversity, and it forms pretty much the preponderance. Here's US data from a while ago. This is now asking, in all the adverse childhood experiences you look at, what fraction of that falls into these categories. You actually realize that while the attention tends to be given to the most severe examples, this is one of the most common examples of early adversity, which is neglect. And when you factor that in, you need to remember that while people are scoring for adverse childhood experiences, it's not the only ACE in the picture. The other ACE in the picture is adverse community environments. And the two of them interact extremely strongly to shape outcomes. So there's an intersectionality between these two, in that if you have a positive community environment, but you have adverse childhood experiences, you may offset some of the consequence of early adversity in terms of individual adverse experiences, because community environments pitch in to offset some of that, whereas it can be greatly worsened when you have both a combination of adverse childhood experience and adverse community environments, and one of the biggest adverse factors is poverty. The other big one is discrimination. So when you start overlaying these, it has a direct consequence on health outcomes as well as learning outcomes. So I just want to bring that up only because, in general, biology replicates this in terms of animal models and understanding it, and does a really poor job of replicating this in our understanding of the consequences of this at a more molecular, cellular, structural, architectural level. It's a little difficult to really model this as well in animal models. So a lot of the literature, at least in animal models, tends to draw from this and not as much from this. But this is a big thing, and there is one more factor that we should overlay on this, which is now emerging as a serious factor, which is climate change. Not counting war, which is another major factor. So disruption of life around you has a profound effect in terms of worsening adverse childhood experiences. We're all witnessing this right now because we're living through war conditions, not personally, but certainly we are experiencing it in the world today. So you can imagine that overlaid with what is going on. How does one then look at the long-term outcomes of this? I just want to once more say that life history carries all the marks of steep inequities in society. So when you look at early adversity, again, data coming out here from the US, where you're looking at race and ethnicity disparity, the likelihood of adverse childhood experiences is higher based on race. And that's not surprising, because you have systemic inequities associated with race in the US. I bet if you look at this with caste data, this is going to be true for India, 101%. It will be true with reference to multiple other systemic inequities that exist in our societies. For each society, that might be slightly different, but it's important to keep in mind the intersectionality of this, that adversity is not experienced identically across different populations. Of course, poverty is a major factor. Discrimination is the other major factor in terms of this. And so when you're looking at the target population for your interventions, or if you're looking at STEM education, etc., for this, you need to be factoring in the barriers and the enablers that will be unique to the population that you're looking at, right? And so this becomes kind of interesting because cognitive intervention / an attempt to look at STEM education for adolescent girls. Here's data from adolescent girls in SC/ST backgrounds. It'll be totally different for an adolescent girl than an adolescent boy in the same background. So the minute you bring in that intersection, you see a very different set of enablers and barriers that get added in the minute gender becomes one additional factor in that mix. I just want to raise this only to bring to the table the real intersectionality and complexity of this problem, and then I'm going to simplify it and tell you what biologists in the laboratory can or do, what do we do with it, right? Like, how do we study this in terms of what is actually happening to the body and the brain? Perhaps the most drastic and dramatic example of these steep inequities was obvious during COVID, when you were forced to witness at one shot what was going to happen to educational outcomes of children across this country because everything went online. And just in terms of who had online access, totally varied based on where you were, whether you were rural or whether you were urban, depending on which state you lived in, etc. And this is just data where this is looking at what the regional disparity of access to internet network strength / a computer. If you just think about this and imagine that the next 30 years is going to involve a large component of digital education in the STEM disciplines, then it's instantaneously obvious that inequities will steepen much more drastically because of access and problems with access. So that's important to factor in, because when one is looking at learning outcomes, when one is looking at what the consequences of this are going to be long term, one will have to factor this into the picture. So I'm going to make a slightly provocative statement. I don't know if it's true. I would be very happy to be corrected if I'm wrong. I'll tell you what scientists tend to do, and then I'm going to make a provocative statement about educationists, which is that we tend to focus on this, the individual. When we're looking at the consequences on the brain, we're looking at relatively genetically inbred animals, asking about the consequences of early adversity in our model system, asking about the impact on that one animal or one model organism or whatever, right? This is your study subject. The problem is that also holds true sometimes when one is looking at this in the educational space, in that you're looking at the individual to some extent and the impact perhaps little bit to the microsystem. The microsystem is school, peers, neighborhood, faith, health services, family, and the way this interacts is the mesosystem. But this is strongly influenced by the exosystem and the macrosystem in Brenner's ecological systems theory, plus what is called the chronosystem, where we sit in time. If you were a girl in the 1960s and you're a girl in 2024 in India, you are in a different landscape. Also, where you live in India, whether you live rural versus urban, your landscape has drastically changed. This is true across the board, and no two individuals live in the same India. No two individuals live in the same Mumbai itself. That itself tells you the degree of diversity we have and the scale of the problem one is facing. But there's a role for science education at all levels, because the least studied is the macro system. So for example, let's just say social policies, cultural attitudes, ideologies, and taxation laws. In today's India, your identity could become a major factor changing how all of this impacts the individual. And that's kind of vital and important for us to think about, because when you're looking at STEM outcomes and you're looking at STEM outcomes for people from different backgrounds, ethnicity, religion, etc., you have to factor this in. You can't wipe this out in erasing information about life histories and erasing information. Everyone who's receiving this is not the same. They carry their life histories, and so whatever you are attempting to deliver is not going to land on the same kind of brain with the same kinds of situations and the same kind of challenges. Okay, so I want to put this provocative statement of Michael Sandel out, which I really like, which he talks about in The Tyranny of Merit. He says, meritocracy is not an alternative to inequality. If you think about it, it is a justification for inequality. Because justifying in a way that almost invites those on the top to look down on those who struggle, who haven't prevailed because they must not have had the effort, the drive, and the talent to succeed. And this perhaps is the biggest problem we currently face, which is a meritocratic hubris. It is the tendency of those who land on top to believe that their success is their own doing, the measure of their merit, and by implication that those who struggle, those who are left behind, must deserve their fate as well. Because in a bottom line, what it says is that you didn't land, I mean, you could say I landed here because of the privilege I carry, or I could say I landed here because of the brain I have. And what you realize is the only reason you landed here is because of all the privilege you carry. And that privilege you carry is huge. And it cannot be erased in terms of the consequences that flow to you as a direct consequence of that privilege that you carry. So to talk about a meritocratic structure without a willingness to talk about the privileges that are so steeped into our society is perhaps a completely, we're not going to make any progress as far as this is concerned. I like this book. I think it's provocative, it's interesting, and it really, it's very interesting in the way it describes some of the problems that currently you see in terms of inequities in society, certainly in the education space as well. Okay, so now I'm going to come to what I'm most comfortable with, which is rodent models, in which I'm removing all those external layers of noise and moving to a relatively genetically inbred system in which we can look at effects of early adversity on the brain and the body. But because I have spent the last 10 minutes telling you how complex the problem is, please do not extrapolate anything I tell you as a straight cut copy paste to the human situation at all. If anything, let this be information to inform you that even when genetic systems are relatively controlled and you can still look at the scale of the problem, there are very many things we still need to understand. Okay, so we know from psychology studies as well as epidemiology studies and studies by psychiatrists that early adversity has a direct consequence on adult outcomes. This has now been studied in many different model organisms, spanning from the fruit fly, Drosophila melanogaster, to mus musculus and Rattus norvegicus, so these are the rat and the mouse, to monkeys, with an attempt to understand what happens to the brain and the body when the individual has an experience of early adversity. And that's what I hope to tell you in the next half an hour. So my lab is fundamentally interested in the circuits in the brain that drive emotional behavioral responses. We want to identify the structure of these circuits, how they talk to each other, and we're very interested in how they're influenced by two things, one the environment and the other by drugs. So in a sense, we're interested in identifying these circuits and their response to conditions in the environment as well as to things that are used to treat psychiatric disorders. And today I'm going to tell you about how early stress can influence these circuits. We study this at multiple levels, from the molecular, cellular, synaptic circuit to the network level. And we're also, of course, interested in how psychedelics work. Okay, so let me tell you now, let me give you a little bit of a primer. And then we'll dive into the data. And is it okay to ask people to interrupt me? Okay, please stop me if I say anything that sounds like gobbledygook and you need clarity. I would much rather have my audience with me and staying with me than you phasing out and falling asleep. So interrupt if I say something that is not clear. So when we evolutionarily designed a gorgeous stress response system, okay, there's something called the hypothalamo-pituitary adrenocortical axis. It's responsible for the secretion of the stress hormone cortisol, if you're human, corticosterone, if you're a rat. Okay? Now, this is beautifully designed for you to cope with any acute stress in your environment. So imagine, let's move back to the era of thousands and tens and thousands of years ago, where you were wandering around hunter-gatherer society perhaps, and you could have become prey to some lion or tiger running after you. You needed to run away fast or escape quickly using whatever capability you had. At that time, you don't have time for digestion, you don't have time for reproduction, you don't have time for even thinking, you just need to move. So all your glucose needs to be metabolized and sent straight to your muscles. So you are reorienting your body to respond to acute stress. The hormone that helps you do that is our glucocorticoids or cortisol. That's secreted by the adrenal glands. They sit on either side of your kidneys, and this is an absolute essential for survival from an acute stress. But you also need to switch off this stuff, because imagine if you keep sending all the glucose only to your muscle and then you shut off your digestion, you shut off your reproduction, you shut off and reorient your resources in the brain. You can't be doing this forever, and also either you're going to be eaten by the lion or you will have survived. And those are relatively fast outcomes, right? One of the two is happening. You lived to tell the tale or you were gone. So there were only two options available for you. So this system was responded to give a very fast peak and then come back quickly to normal and allow you to proceed with whatever you wanted to proceed with. You don't want to switch this on chronically for long durations of time. So the system also has a beautiful feedback inhibition axis in the brain through a structure called the hippocampus. Also turns out that that's the structure that's involved in learning and memory. It's the absolute essential component of the brain that's vital for acquiring and learning new explicit memories. So the hippocampus talks to the hypothalamus, shuts off the hypothalamus, which tells the pituitary to no longer secrete whatever hormone, doesn't matter the name of the hormone, to actually cause the adrenal gland to secrete corticosteroids. And this happens in a nice feedback loop. These glucocorticoids bind to glucocorticoid receptors and mineralocorticoid receptors, some of which are present in the hippocampus. They tell the hippocampus to switch off this particular pathway. So this is a nice feedback control on the system. And you do this to switch off the stress response. Now, you also switch this on in chronic stress. Let's say there's a famine, or let's say there's a very bad flood, or there's some sort of there's drought, etc. These sort of conditions will also require you to switch this on, but you're switching it on for longer. You're switching on the same thing when your boss has not given you a promotion. You're switching on the same pathway when, let's say, you're on social media and somebody didn't like your post, or someone was really mean to you, a social stressor. Same pathway being switched on, except unlike the lion who will either eat you or you will escape, this doesn't end, especially if you're chronically switching it on, and these are psychosocial stressors. So what you have is a circuit that was beautifully designed but has got hijacked by circumstances that have moved faster than our bodies can evolve. The world, in terms of its environment, changes way faster than the evolution scale of these pathways in your body. That is moving over tens of thousands of years. So now this pathway is being switched on if you had a fight with your neighbor who has perhaps parked a car in your parking spot, and the two of you have been fighting, let's say, every single day for months and months. You see that person, you get a full-fledged cortisol spike, your body is in shutdown mode, your digestion is shut off, everything else. You hate your boss, same pathway. Okay? So this pathway is now beautifully hijacked for something you have no control over and you can't switch off. So this becomes a serious problem. And in individuals who go through early adversity, this entire pathway gets primed to prepare for future stressors, with the view that your early life is perhaps a testing ground for you to predict your future. And so if you have high adversity and high stress, you respond in terms of the circuit by having a hyperactive, hyper-vigilant HPA axis. It's on. It doesn't switch off as quickly. You get very strong responses and it runs away with you. So you end up bathing the body with this cortisol for a lot longer. Okay? This is a serious problem. As a consequence, you end up killing neurons or changing their architecture, or changing their shape in structures like the prefrontal cortex and the hippocampus, key components of emotional neurocircuitry. Okay, so all this was understood. Now, what are we doing right now in our lab at TIFR? And I have the pleasure of talking about Prateek's work. He's right here. So he's an early career fellow, India Alliance early career fellow in my lab, and it's his work that I'm going to discuss today. So we've known for a while that aging evoked neuronal damage and cognitive decline is speeded up if you have a history of early adversity. Early adversity is not only a risk factor for psychiatric disorders, it just shifts the axis on the entire aging process. But how precisely it does it and what's actually happening in the brain is perhaps not as well understood. All right, so this is the window we are interested in. And I'm now going to switch very quickly to talking about rats. So all this will shift in terms of its 2.5 years is a very, very old rat. Early life is the first 3 weeks of life. So this is now going to move to rat models. Okay. So what Prateek wanted to ask was, does early stress trigger inflammaging, which is, does it drive up inflammation in an uncontrolled manner which speeds up the process of aging? That was the question. And associated with that is there dysfunction of the powerhouse of cells, which is the mitochondria, which is essential for keeping all cells functional and active, but is particularly important for neurons, which are the only cells you have with you for your entire life. Everything else is turning over and replaced. Neurons you want to keep, and you want to keep for your rest of your lifespan. So their energetic regulation and quality control of mitochondria is way more important than perhaps your skin cell, which, for example, you're shedding every single few weeks, right? Also your bone, you shed and you turn over. Okay. So what I will do is now tell you the precise kinds of experiments Prateek did. He looked at a model that is a model of maternal neglect. It's one of the milder models, and I'm saying that specifically because more severe models just shift the entire thing even faster. They hasten it even more. This model is based on taking the pups away from their mother for 3 hours every single day from postnatal day 2, 2 days after their birth, till postnatal day 14. So for 12 days, every day they go away from their mother for 3 hours. First and foremost, let me please tell you that do not use this and cut, copy, paste and extrapolate this to humans, because this will not apply at all. It doesn't even apply to monkeys, because this becomes a concern, because everybody then says, what are you saying, women cannot leave their children? Not at all, far from it, right? Primates are much more complex caregiver societies. It does not work if you separate the pups from the mother. You have to separate and isolate them from all peers. Remember, there are aunts, grandmothers, fathers, uncles, there's a whole complex caregiver society. So for primates, this doesn't work. If you do the experiment with rodents, where there is a strong paternal component, then deprivation from the paternal component also evokes this kind of a stress. In this species, in this model, there is no paternal component, which is why the caregiver here happens to be the mother. But it is essentially a model of caregiver neglect. And when you move this up and you expand this to other models, etc., humans, you need to remember that it's a complexity of caregivers and not just the mother. I want to make sure that I say this very clearly, because I don't want the people walk away with this misunderstanding. Yeah, or no? You can define it in multiple ways. You can look at it as lifespan and the natural process, and every animal house will, for example, have a slightly different timeline depending on what their animal house conditions are. You can also look at telomere length, for example, in peripheral tissue where there is active turnover, where telomere length is, so, you know, shedding off the tails of your DNA, the telomere, attrition of the telomeres is a marker of aging. And people have already shown that individuals with severe early adversity show telomere shortening. So that has already been seen in humans. So we know that that speeds up. Yeah, though you can use many senescence markers. So cells, as they get older and senesce, have a battery of changes that they show. And you can look at these at the molecular protein network level. Yeah. So what Prateek decided to do was ask, okay, I will take this model of relatively mild maternal neglect, because it's a milder model, and I will look at these animals when they are adult and middle-aged. I will focus on the hippocampus, because that's a structure that's so central to control of the stress response axis, And I will ask what happens to mitochondrial function, neuroinflammation, peripheral inflammation, other aging-associated markers, blood-brain barrier integrity, and cognitive behavior, and try to see if there is a relationship between all these things. So what Pratik did is, here's the model, okay? So there's the dam, that's her pups. When they are born, they get taken away every day for 3 hours, and then they go back to their dam, and they are usually taken away as a cohort. There is, of course, no real serious nutritional deprivation in this because the animals seem to be feeding fine. The number of nursing bouts is similar, but the nature of nursing bout is completely different. The dam switches, because you do this perturbation in her environment, she switches to being a passive caregiver with far less licking, grooming, and arch-back nursing as compared to dams that are left undisturbed. Okay? So it's a model in which maternal care shifts. So when you do this and you look at them at 2.5 months, 6.5 months, and 15 months, and you measure C-reactive protein, how many people in this room have had CRP measured, HS-CRP measured in their blood? I'm trying to make sure my audience is awake. So just testing. Anyone ever had a CRP measurement happen in their blood? Somebody must have. Or you're just, this is the classical thing all doctors look for to ask if you're carrying inflammation load in your body. CRP is a good marker of infection history, inflammation, etc. So you see that at 2.5 months, really not much going on, whether you had a history of maternal separation or not. By 6.5 months, there's a small increase. At 15 months, there is this really severe cumulative inflammation that shows up in animals that had early adversity. You should realize that the axis itself is changing, so it's not like it goes down. It's actually going up, but so this is much higher than what it was at 2.5 months. So regular healthy animals actually show an increase in and of itself because, yeah. 15 months is middle-aged. Yeah, so in middle-aged life, yeah. Yeah, so you go 6 hours, you'll start nutritionally depriving these guys. People have looked at this paradigm now for quite a few years. You don't want to get them cold because they're not thermoregulating when they're very small babies, and you don't want them to miss too many nursing bouts because then it's a nutritional deprivation. 3 hours, there's no real nutritional deprivation at all. They get the same amount of nursing bouts, and they're not cold because we are keeping them nicely thermoregulated on a heating pad. They all go together. If you take each animal out individually, it becomes a very severe paradigm. So there are paradigms like that where each animal is isolated and taken away. That's a severe paradigm. The whole cohort goes out and it comes back within 3 hours. Yeah, there are many other versions of this that have been that people look at. There's much more severe paradigms. We were particularly keen to use the milder one on the spectrum because we were trying to model not severe neglect, but neglect. Yeah? Okay. So now we were looking in the serum in adult animals at 6 months of age for inflammatory factors that you associate with inflammation. IL-1 beta, IL-6, IL-18, tumor necrosis factor. Some of these names might be familiar to you during COVID. If anybody had COVID and their doctor was vigilant, they were testing for some of the interleukins. Okay, CRP was standard, but also the interleukins. And what you see is at 6 months, not much is drastically changed. IL-6 and tumor necrosis factor are normal, but IL-1 beta and IL-18 are already up in both males and females. And obviously, each individual spot is an animal, so it gives you a sense of the heterogeneity. There is heterogeneity in the response, right? And that's not surprising. That's what we are interested in also capturing more. Okay. So now what happens if you wait till 15 months? So these are animals now that are undisturbed out till 15 months after that history of early separation, and now you're looking at their serum, IL-6, IL-1 beta, tumor necrosis factor all up. So all the pro-inflammatory cytokines go up, anti-inflammatory cytokines like IL-10, IL-4 go down. So you're tipping the balance to pro-inflammatory and reducing the anti-inflammatory. So the protective factors are going down, the worsening factors are going up. Okay. So now the question is there an associated mitochondrial defect in the hippocampus at that same point? So what Pratik did was measure ATP. ATP is like your currency of energy in the cell. So that's what the cell burns to do its work. You need ATP to actually do the work, and so you're really measuring, in a sense, your, I guess, your rupee value as it is changing, or your stock index as it's modulating, right? So you look at ATP levels, nothing at 6.5 months. By 15 months, you see a decrease in both males and females in the hippocampus. And if you now take out mitochondria directly from this part of the brain and measure oxygen consumption, which is you're measuring how much respiration these mitochondria are doing. What do mitochondria do? They take the oxygen and the glucose that you have eaten and the oxygen, and they use this to burn and generate. Essentially, it's like a combustion system that is giving you energy at the end of it, okay? So you're asking, if I give different components and block specific components of the electron transport chain, what is happening to my ability to do cellular respiration in the mitochondria? The red line is maternally separated. The bottom line is you are not getting, the mitochondria are not functioning optimally. For every bit of glucose that you get, or pyruvate or succinate, you are not utilizing it most effectively and optimally. You are getting far less out from whatever investment you are receiving, okay? And you see that that being measured out as well, okay? Along with this, so cellular ATP, as I told you, is down at 15 months. You're building up reactive oxygen species. The side effect of doing this cellular respiration is you also produce oxidative free radicals, and the free radical buildup starts happening as well in the hippocampus. Okay. Let's look at the direct, so all this points to few things. It points to the fact that slowly but steadily, if you leave these animals with a history of early adversity, a mild model alone, 6 months there are some changes but not a lot going on. But as they age, you start seeing this worsening profile with inflammation and mitochondrial dysfunction both showing up. Now the question is, what is the hallmark indicator of inflammation? Can you find inflammatory cells? Cells that are actually associated with inflammation in the brain. The cell that's associated with this is microglia. And microglia, you can actually look at the way they look. So here are this is what they look like when the brain is in a healthy, good state. As they go into really having a lot of inflammation, they become like this. So they change their architecture and become strongly amoeboid, and these are the ones that are chewing up all kinds of stuff, including stuff you don't want. That means the branches of your neurons, chewing that up, phagocytosing, things that you don't want removed, they're just activated and removing things you don't want as well. And what's obvious is nothing is happening at 6 months. Your control and your maternally separated do not look any different from each other. But at 15 months, you're beginning to see the appearance in multiple subparts of the hippocampus of these activated microglia telling you there is this increase in inflammation. Okay. Another way to look at inflammation is to look at the inflammasome. Suffice this to say, just think about this as many molecules that come together to form this big complex which drives the inflammation response in a cell. So you can actually look at these molecules, and when you do that, you can see that here are control. So this is the molecule NLRP3, this is ASC, doesn't change in adulthood but goes up in middle-aged maternally separated animals, which you can quantitate here. Okay. So what have I shown you so far? Early life stress results in a slow, cumulative, gradual increase in inflammation and mitochondrial dysfunction that appears slowly in the periphery. What is going on in the brain and the muscle, for example, or the liver? I told you about what's happening in the periphery, and in the brain I've also told you that what you see are changes at 15 months, but nothing going on at 6 months. What does it look like in other organs, like the skeletal muscle or the liver? What was remarkable is that the body starts keeping score before the brain does. No. At 15 months, not at all. Very clearly, both the male brain and the female brain, both the male periphery and the female, similar changes. Yeah, yes. Can you wait and hold that question, because that's the million-dollar question. Can you reverse it? Yeah, absolutely. What I want you to know is that if we were to look at another organ like the liver or the muscle, the changes come a lot earlier than the brain. We actually see changes at 4 to 6 months in the liver and in the muscle. So that tells you that the body starts keeping score even before the brain starts keeping score. And that's just to show you some of the data. We'll ignore that for the moment. Okay. So then something interesting is going on. You're privileging the brain. You are protecting the brain at the cost of other things. You already have lots of peripheral inflammation that starts early, but it's not very rampant till about 15 months of age. The muscle and the liver are already showing changes at 4 to 8 months, but none of those same changes are seen in the brain. In the most vulnerable region, which is the hippocampus, the prefrontal cortex doesn't even show those changes at 15 months. That might come perhaps even later. So the question then is, how are you privileging the brain? The natural question is, is it your blood-brain barrier which acts to keep out all this active peripheral inflammation that's going on in the body and protects your brain? Here's what a healthy blood-brain barrier would do. It would clear out junk or not let it in, first of all, because it just throws it out. It's a physical barrier, and it's a physical barrier to immune cells going in, right? Like, they don't just enter the brain. Remember that if you have injury in the brain, you've squished a lot of material into a small skull. It's a problem. So which is why when injury happens and fluid buildup happens, there's no place for it to go. It's a design flaw by definition, because there's really no space for any of this fluid buildup or immune buildup to act. Imagine what happens when you smash your finger and it swells up nicely. You cannot do that easily in the brain. There's no space for this kind of stuff to fill in and swell up. So you don't want too many immune cells entering your brain just like this and moving around in all places. So blood-brain barrier is critical. And if it breaks down, what's going to happen? So this is what Pratik decided to ask. He looked at the building blocks of the blood-brain barrier. This is looking at gene expression. Forget what the individual names are, not important. Each of these are structural components or other tight junction components of what builds your blood-brain barrier. In the adult animals, so green is the maternally separated, black is the control. You can see nothing has happened at 6 months. Your blood-brain barrier components are not changed. But if you look at middle-aged 15-month-old animals, lots of components are already beginning to go down. So it looks like, at least at the gene expression level, what you're building as your barrier is beginning to start deteriorating. But the proof of that would be if you can actually measure the strength of your blood-brain barrier. And one way to do that is to use a dye which should be large enough that it is excluded by the blood-brain barrier. It should not go in. So if you look at the brain, you should not detect the dye. It's an Evans blue dye, right size, some X amount of kilodaltons, I don't remember off the top of my head, but appropriate size that is normally not going to enter the brain. Sure enough, in control animals and maternally separated animals at 6 months, if you measure the amount of Evans blue within the brain, there's really nothing going on. But look at what happens in a middle-aged animal. It's like you converted your blood-brain barrier into cheesecloth, where anything can now start going through. So this molecule is now entering the brain tissue effectively because your blood-brain barrier is not intact. You've compromised your blood-brain barrier. This is a bit scary because this is a relatively mild stress, and this is a consequence you're seeing months and months later, but the fact is that it tells you that early adversity compromises the blood-brain barrier and makes it kind of a little bit like cheesecloth over a period of time. As I said, more severe the stress, you will move this forward, right? So this is still relatively milder stress. So the main findings from Pratik's work have been to show that in adulthood, the changes are really in the periphery and in organs like the liver and the muscle. But as you get to this window, all that has been happening in the periphery now seems to have access to structures like the hippocampus because you have made your blood-brain barrier kind of like a cheesecloth. So things are going in, and you have access to inflammation now in the brain. All these inflammatory molecules can go in. Is there an association between all the different things? I didn't tell you what is happening to learning at all. I didn't tell you about cognition. So is there a link to, for example, learning? And is there a cross-correlation between all these things that we are measuring? So here are 15-month-old rats tested on a task called the novel object recognition task. Very simple task. Rats like to explore objects. So on the first day, let's say you give it 2 circular objects. So it will spend time exploring the objects and it enjoys exploring them, and it's a nice little new novel thing for it to do. And so usually first we habituate the animal to the box in which the training and the testing is going to be done. So the box is not novel anymore. They've seen it for 3 days consecutively. On day 1, they see these 2 objects. On day 2, on the short-term day, we replace one object with a new object. Perhaps like, let's say this is a beaker. This could be a ball. Yeah? So they had the same old beaker that they saw on day 1, and now this has been replaced with something else. So you're asking the animal, can you tell that this is new? They would prefer to spend time with something new. So the ability to discriminate that is what you're testing. It's a novel object discrimination task. So you have a discrimination index. A positive index means that you could tell what it was, and you're not spending equal time. Equal time would mean you'll be at 0. You don't really care which one the object is. So these are 15-month-old rats, and they, of course, prefer to spend time with, and this is 7 days later, actually 5 days later from this, so this is day 7 of testing. Again, the same familiar object and one new, some completely different object. And you ask, can you again tell that, hey, you saw this once, twice, and now you're seeing it the 3rd time, and this is for the 1st time. So can you do this in both a long-term and a short-term novel object discrimination task? Control animals do pretty well, though there are some controls that don't do well as well. Okay, so important to know that there's a distribution. But when you look at maternally separated animals, you have a larger cohort of animals that are doing worse. Now your question is, what does it look like across the distribution? Can you start asking if this is correlated with any of those peripheral things you have looked at? And so what you can see is it's very strongly correlated with inflammation. The lower your discrimination index, which means the worse you do on your learning task, the higher the amount of inflammation you are carrying. And that's true with IL-1 beta and tumor necrosis factor as well. And IL-10, which is an anti-inflammatory, the lower your anti-inflammatory, the worse you do. So basically, more the inflammation, the more the strong correlation with a poor performance. But what's interesting is this holds true only for the maternally separated cohort, because when you move to the control cohort, I'll just show you the data together. Here are the red line is the maternally separated animals, and the blue line is the controls. And our working hypothesis is that the controls at 15 months, the inflammation, first of all, hasn't built up that much. 2, they're still middle-aged. They're not aged. And our view would be that this axis, this will move like, this will steepen in this direction as these animals get older. Whereas the maternally separated animals, which are already showing an accelerated aging phenotype and a cognitive decline, are showing the steep graphs. This is interesting. This is something we're testing more closely to look at. Let's come back to Yasmin's question. You have this big window between the time you did that early adversity, and then, of course, this is an artificial situation in which no other hits are coming other than the hit of aging. Aging is itself a cumulative stress, but nothing else is happening to these animals. Can you rescue this at any point, right? So we got funding from this from a philanthropic organization. It's very interesting. It's a doctor couple based out of Telangana who are donating their entire personal wealth to science and scientific research. And we happened to get their first grant when they gave it out, and they are very involved in a review process. So every year you review whatever you found, and they have a very nice panel. And in the review discussion, one thing they said to us is, we know you'll want to look at serotonin pathways and inflammatory pathways and all of that, but can you do something instead with something that could be over the counter and that doesn't require FDA or drug administration, drug controller approval, etc. Can something that could be translated much, much more easily? So that was a very clear mandate of the funding organization. So they said specifically, we want you to look at nutritional regulators potentially as in. Then we had to scratch our heads, and we spent a lot of time thinking, what do we intervene with? What is it going to be that we can come in? And there were many candidates. Factors of cost were a factor. How easily will we be able to give this to the animals? Is it potentially something that's cheap and has been used extensively? What can we do, right? So in the process, we narrowed down on a molecule called nicotinamide, which is vitamin B3. Okay, vitamin B3, by the way, just to be aware, most people have B complex deficiency in our country. So that's another intersectional factor to take into consideration. This is vitamin B3. It's a direct energy source in a sense because it provides you reducing equivalents for your mitochondria. It's been suggested as a direct mitochondrial booster. Mitochondria often are the first seeders of an inflammatory situation. So this became a candidate for us to try. And so what Pratik decided to do, and he has many of these experiments going on, we did maternal separation, one where we were just repeating the same experiment but with regular drinking water, which they had anyway through the whole time. Or we gave them nicotinamide in the drinking water to drink for a period from 8 months to 13 months. So they were taking it for a period of 5 months. I don't know what that was. Okay. Here's what he found. So now you're looking at CRP, GDF15, which is a major factor associated with aging, methyl glyoxal, which is again another one of these senescence factors associated with aging, corticosterone, which is the stress hormone, and cell-free mitochondrial DNA, which is a factor that is secreted associated with stress. What you can see is CRP is up. We're doing something here which is really interesting. There's a shifting downwards. That's also true of GDF15 to a certain degree for methyl glyoxal. Corticosterone, we're bringing this hyperactivated stress response axis down. Cell-free mitochondrial DNA, again another measure of stress coming down. This is super interesting to us because it tells us that months and months after that history of early adversity, it's possible to come in with a modulator and potentially change some of these outcomes. Inflammation, or the hallmark of inflammation, are those activated microglia. And what you can see is that this activation of microglia in the hippocampus in different subdivisions is being attenuated in animals that had this combination of nicotinamide with a history of stress. So here's nicotinamide given to controls, here's nicotinamide given to maternally separated animals. This is control and maternal separation in regular drinking water. And what you can see is that you're beginning to reverse some of these signatures, which is fascinating.
Why does it go up for the control? I mean, it doesn't seem to be good for people who...
This and this? Yeah, I don't know. I mean, sometimes we see that. It's not really a significant effect. But you do see variability. I mean, that you know, we are going to have to do this with large-scale animals. And this is without bringing in generic heterogeneity as a major factor. So when you're putting this data over to the human, that's a big concern that you'll have to keep in mind. Here's ATP levels. You can see a decline in ATP levels. You can see this increase in reactive oxygen species, and that starts getting attenuated. Lots of experiments still ongoing. We need to look at this much more closely. But this is perhaps the most interesting data. So when you look at the blood-brain barrier associated genes, they are reduced. So here's control animals. These are all the building blocks of the blood-brain barrier, and it's normalized to the control. Here are maternally separated animals that had normal drinking water and waited till 13 months, and you're looking at the blood-brain barrier. The blood-brain barrier level expression of many genes is down, which is why it's expressed in blue. Here's control animals on nicotinamide. Here are maternally separated animals on nicotinamide. And what should be obvious is you're rescuing gene expression for many genes. And that's super interesting. He's actually measuring the blood-brain barrier. So once we know that, we'll actually know the porosity of the blood-brain barrier. Here's the data as far as cognitive behavior is concerned. Here are control animals on drinking water, maternally separated animals on drinking water. You have a cognitive decline. Here's control animals on nicotinamide, and here are maternally separated animals on nicotinamide, telling you that you have some degree of rescue. The fact that you have any rescue at all is very interesting. So this is where I'll wrap up and end and say that, you know, when you're in a basic science lab, you tend to ask the question by trying to take away the noise. You have a model system in which you have genetic heterogeneity reduced. You ask what is the effect of early adversity to ask what happens to the brain and the body. The reason I started with the discussion about how complex it is to extrapolate this is because there are so many factors at play when the minute you switch over to the human. But you need to study this to understand what potentially can go wrong. So this has been led by Pratik in the lab, who has been an exceptionally talented and wonderful colleague to have. Nishta Pange, who is a research associate who has worked very closely with Pratik, and many other people in the lab as well. I am not going to acknowledge all of them, but I would definitely like to say a big shout out to Sheetal Suryavanshi, who heads our animal house. It's non-trivial to do these experiments across an entire lifespan. My collaborators, my father, Ashok Vaidya, Ullas Koltur, who's a mitochondrial physiologist at TIFR, and Professor Carmen Sandy at EPFL Lausanne, who's a very close collaborator of ours on this project and has been deeply involved in the choice of looking at things when we're looking at reversal. My lab is actually right there. It doesn't even show up, but it's right here. The Hutment lab is at actually literally almost on the coastline. That's our logo. You're more than welcome to come visit our website, come visit us. That's the team, funding from TIFR DAE. This grant, which I would really like to acknowledge, the Sri Ramakrishna grant from the Padmavati Venkateshwara Foundation, which is a philanthropic grant. It's been remarkable to have that sort of philanthropic support to drive some of the science, a Fulbright Fellowship, India Alliance, that's the Wellcome Trust and DBT. And I am done. I'm happy to take questions.
Thank you, Professor Vaidya, for this talk. And let's see, actually, first, let's take some questions from our audience here. Okay, while they prepare, maybe I can ask one. So I'm just curious, like, what happens, let's say, due to some unfortunate circumstances, there is a memory loss. Does brain gets a second chance to train how to react to stress kind of situations?
I mean, we have memory losses all the time because forgetting is an equally important process for the brain as much as learning and remembering is. In fact, forgetting is a vital component of the nervous system. So when you lose the ability to forget, that can become a serious issue independently. Routinely, we are dropping things that are not salient or not important to us. I think the brain is a salience device. Its job is to remember what matters to you. It's not wasting time on stuff that is irrelevant. So a lot of irrelevant stuff is just being shed on a daily basis, which is you're weeding out information, picking that which matters. And so anything that increases, so if there's a memory that's not a strong memory, but there's a strong salience to you being able to recollect it, it will come straight back up. Every time a memory comes back up, it is in principle available to rewrite, which tells you that nothing you actually remember in terms of explicit information is actually ever the true fact. It's just rewritten because you've opened it up over and over and over again to rewriting. So especially for explicit memories, and that's why one of the things the justice system talks about is the importance of getting the facts at the beginning, because after you've talked to 4 people about it, the facts change. They just change in retelling over and over again. They become different as they go along. So, yeah.
Okay. Hi, ma'am. So my question was, when you did the cognition test for mice, when you were checking how they respond to new objects, did you all check whether the mice follow the edges of the box to go and check, or it goes in the center?
No, that's a good point. So our objects are not in the center because the center is the zone that the animal likes to avoid because it's a little nerve-wracking to be in the center of an open field. So they're on the side. But we have independently checked that multiple times, and we know that these animals show anxiety increases when they are young. Actually, in the first in that 4 to 6 month window, actually 2 to 6 month window, they have a higher amount of anxiety. But as they age and when we get to middle-aged life, we see a lot more of cognitive deficits than we see anxiety. If we don't do anything to these animals after that, the anxiety eventually weeds off. So we don't see the sustained increase in anxiety. You need to come in with a second hit to see a very sustained effect on anxiety. So no, they don't have an anxiety effect.
Yeah, Meena.
Hi. Very interesting work and talk. Thank you for that. I just wanted to know, you mentioned cohort, right? You are separating the cohorts of the babies. So I was just wondering, maybe, sometimes cohort acts as a supportive caregiver, to like siblings. Yeah. So even then, you got these results. And then I was wondering, if you change the model, then would you get solitary. That was one thing. And the other thing is...
Should I answer that one first?
Okay, I have second one.
Yeah, I'll come back. So yes, absolutely. When you separate each individual pup separately, it will get worse. A cohort is itself a component of this experiment because some cohorts might buffer better than other cohorts. And so we have to constantly make sure we have multiple litters. We are not looking at one litter. We're looking across multiple dams with many different litters, because litters become, so the entire cohort may buffer better than another cohort. So that's a factor for sure. And people have done that experiment where they have separated each individual pup. That's actually quite severe. And you see much more severe effects even when they are young. So
and the second thing is, anyway, we are seeing stress building up. So I'm just wondering, what are they prepared for actually facing stress from some other factors?
I love that question. So we published a paper years ago now where we showed that these animals actually do much better on stress-associated learning tasks when they are young adults. So there is an adaptive component to the stress, in that it's almost as though the experience of early adversity helps you to do far better when the task has a stress component and you outperform controls. But nothing is like, there's no free lunch. So there is a cost to this adaptation. Yes, in that early window you adapt, but then there's a cumulative cost which shows up much later, but it does show up. So we did write about this as the fact that it's like there's no free lunch. You have a cost to pay for that adaptive, and there is a big adaptation. So we were quite surprised by this when we first found it. It was a paper that we published in 2013 where we were like, these animals are just learning better. What is going on? They outperformed the controls on learning tasks, but the learning tasks had to have a stress component, not on neutral tasks. If you did it on a neutral task, they were just like the controls, nothing drastically different. But on a stress-associated task, they just outperform the controls completely. So yes, there is a, and that makes sense, right? It's like your body is attempting to adapt most effectively to the history that you have and to utilize that history to the best possible outcome. But there is a cost.
Thanks.
Thank you. My question is about the blood-brain barrier. Seems like a very complicated concept, but one question I had, you used a line that brain is privileged. So I was wondering whether this blood-brain barrier is made by brain sort of a, so who creates?
The blood-brain barriers.
Can I also ask the connected? And the second one is whether the possibility of artificially, like from a medicine point of view, could you create these blood-brain?
So the blood-brain barrier, so essentially think about it like this. Your blood, what is the barrier for? There is blood vessels going through your entire brain, right? It's highly vascularized, which is why you don't want an internal bleed in the brain. I mean, that's one of the biggest concerns. Small territory, no chance for this blood that leaks out, it causes damage. So you have to prevent things you don't want from entering the brain, from leaving these blood vessels and entering into brain tissue. So the lining of the blood vessel on either side of that has both, so there's endothelial cells that line the blood vessel, and then on either side you have astrocytes that form these end feet that actually sit along the entire wall of the blood vessel and keep an intact barrier so that only things that can be actively transported, for example, that have transporters, or things that actually have a mechanism for, it's like saying BRC needs security to enter. In some sense, this is perhaps the best analogy. You can't go in if you are carrying a USB, if you're carrying a mobile phone, you can't leave all that and then go, right? So you are literally gatekeeping what can go in, because it's an extremely important control center and you want to watch what is entering. So yes, it's made by cells in the brain in combination with the cells that form the walls of your blood vessels. So it's that combination that determines it. But you still want some immune cells to go in. So there is passage, there is ways, but you are controlling and regulating that entry. When the blood-brain barrier collapses, you lose the regulation. So in principle, one concern would have been that when you build that big Atal Setu on the other side, you now provided a vulnerability factor from the other side, right? If that leaks, you have a big problem. So that's the best possible analogy I can come up with. I mean, for things you want to really gatekeep on entry, you need tight protections. And it's made by both the brain and the... Can you rebuild it? Really difficult to pull that off. Think about it as... Just think about it as how your capillaries are in your, they're not, it's not like one nice road. Imagine if you had to rebuild every road in the city of Mumbai. In a sense, this is like rebuilding. I mean, you can see what has happened. Every time we try to rebuild the road, we create another pothole. So we are very remarkably good at that. But to rebuild everything, because it's on every single capillary / artery that is going through the brain. And it's a very vascularized structure, so not easy to imagine that. Yeah.
Hi, Vidita. Thanks. Really nice talk. 2 questions. One is the vitamin B3 supplementation should show up earlier in the circulating peripheral macrophages. And have you checked that?
Not yet. What we've looked at is peripheral inflammatory factors. So it shows up at all the serum factors show up, and you can see that. But we started our intervention at 8 months. And
I was wondering how you knew that.
So we went late based on some other studies we had previously done where we wanted to intervene not too early and overlapping with the early stress. But Pratik has a cohort now that's running where it started significantly earlier. We are stopping the treatment and then we are letting it go. And then seeing whether a short duration treatment earlier with then this big gap after actually still protects. Does the timing of the treatment matter? I don't know. The difficult experiments to do because all these are 5-month interventions and then you have to wait a year to actually see the result. So we have one cohort that's going earlier. We'll see what we see. But I would predict that we will rescue the periphery as well. We have now gone in at a time point where the peripheral changes have already set in. So we are at 8 months. Whether we can reverse a set of changes that happened is a question. We're clearly reversing some of the interleukins and GDF15, etc. So peripheral inflammation markers are coming in. But we can look at this.
I meant more as a marker because then you can try different things.
And now what we want to do is we want to translate some of this to human situations. So we have a collaboration with NIMHANS where there are patients with a history of early adversity versus those without. And then patients with a history of early adversity that manifest with psychiatric disorders or not. So we have all these cohorts and then we will run the peripheral Now that we have markers that we know are strong, we are anticipating a lot more heterogeneity the minute we move to the human, but we'll still look at it to see what we see.
So that brings me to my second question. Is there a converse relation? Like in autoimmune diseases or neuroinflammatory diseases, is there an increased anxiety or...
Oh yeah. I mean, the amount of evidence on this. So there's this whole hypothesis of depression and anxiety and psychiatric disorders actually being disorders of inflammation, cumulative inflammation in the body and in the brain. So there's the inflammation hypothesis of psychiatric disorders. So yes.
Thank you.
Okay, so in interest of time, we'll just do last 2 questions, Mukul and then Anvesh.
So this is a randomized control trial, right? So what are the factors?
It's not a randomized control trial at all. I mean, what we've done is we've taken animals and gone in with an intervention where when our analysis is done, we are blind to the treatment groups. But it's not really a randomized control because then you have to be double blind, which we are not.
Okay.
Yeah.
So, I mean...
When most people don't do double blind trials with animal models.
Okay. So, so while selecting your initial sample, what? You have like randomized selection of mice or it's just
this is where animal models take away this noise, right? We are looking at genetically very similar animals that are bred in an animal house. We will just move them at random into groups, but really there's no big selection criteria, partly because we have been a minimal heterogeneity in our starting population. Our starting population is essentially coming out of the same cohorts of litters. We have, the one way to do it would be to segregate the same dam and then but you can't do it because of the design of the experiment. You can't have the same dam for both. But other than that, the conditions are very, very, very similar. So we randomize in that if we are taking like 20 animals and there are 20 animals across multiple litters, that's it, and for the same animal house. The genetic noise here is minuscule compared to the real world, which is why at the beginning I wanted that to be clear. And that's pretty standard. It gets even less when you move to mice, which are inbred. This is still an outbred rodent line, but it's minimal.
So like another quick question.
Sure.
It's about, so at how often and at what levels these studies in mice apply to other animals, or especially to humans? Can you give like one very good example where such kind of studies have been applied to humans?
I mean,
for
I think
example?
perhaps the biggest example is if you look at defects in the hippocampus, volumetric. So early stress is associated with volume reduction in the hippocampus. It is true whether you're a rat, it's true whether you're a mouse, it's true whether you're a monkey, it's true whether you're a human. Hippocampus volumetric loss is something that's a hallmark signature associated with early adversity across species. Same thing happens when you measure cortisol in humans, cortisol in monkeys, corticosterone in rats or mice. The HPA axis is abnormal and you have higher levels of corticosterone in animals that had early adversity, higher levels of cortisol in humans, failure of effective feedback regulation seen in rats, seen in mice, seen in monkeys, seen in humans. Biology is parsimonious. If something works, it utilizes the same model over and over and over again effectively. It's not wasting its time in reinventing the wheel across evolution. So much of the underlying building blocks will be similar. What will be drastically different is the genetic heterogeneity that will overlay on this. Right? We are looking at, the minute you move to an animal model, you're shutting down genetic noise to a very small extent. The minute you move to a human population, you are increasing your genetic variability on a large scale. So your sample sizes go out of the roof. You need thousands of people with reference compared to 20 animals or 30 animals. It just changes the scale because genetic noise comes in. We are minimizing it. So we don't have that as a major factor. And that's where a big issue comes in. Like let's take an individual, a human being who's carrying, for example, a serotonin transporter polymorphism. They may not respond identically to another individual who's not carrying that polymorphism. Levels might be different, the degree of response or vulnerability might be different. So that is the noise that comes in the minute you move to human. You don't see that in the animal models because you're wiping that noise out, artificially wiping that noise out to understand what actually happens. But in reality, that noise is a huge component of the, yeah.
All right, Anvesh.
Hi, Vidita. Very interesting work and very nice presentation. So you said that these, just now also you repeated that these do not translate to mammals or things like that. But I'm wondering...
I mean, a cut, copy, paste can't be.
Right.
You have to factor that in when you're doing the translation.
Sure. So if I understand correctly, so you take this cohort or this litter out from, separate it from their mother. I'm just curious, have you put them along with a different mother? That's one. And the second is that in mammals or something, we also see the mother do some physical activity with the litter. For example, I mean, the tiger or somebody will lick the cubs and things like that. So do mice do that? And in this case, how do you, because you said that you have already taken care of the temperature control and the feeding, but what other activities might the mother be doing?
Great questions, Anvesh. We haven't done it, but other people in the field have. And they are really beautiful studies where cross-fostering or adoption will override most of the effects of separation. So if you cross-foster, but you have to cross-foster early. As in the early cross-fostering is required for the foster mother to accept the litter. But when you cross-foster, most of the signatures go away. So there's a big, and this has been a big discussion in the world in terms of referencing adoption and early adoption having some advantages as a major intervention, especially when there's been adversity. So that there's a lot of data on that, both in monkeys and in rats. Across mammals, many things hold true and translate. Some things don't always hold true, okay? Now, the second question about whether stroking, right, and licking and grooming, you can actually sit there and measure this. And you see how much time it spends, where is it licking and stroking. You can sit, and people have actually done this work from Stefania Macari's group, where instead of the mother, you take the pups away, but you have graduate students sitting with wet brushes, and they are stroking the pup actively during the time of separation. So in those 3 hours, you have a wet brush and you're just stroking the pup continuously. Overrides a lot of the signatures, which is really interesting. So there is clearly a key somatosensory tactile component that is involved, and that perhaps has been one of the biggest learnings in terms of translation from animal studies to human, from Harry Harlow's work and other people's work, where in the past when you had a baby born prematurely, you would segregate the baby and put the baby in an incubator with minimal contact, tactile contact with either the mother or with any other adult. That has actually been overturned because the stress associated with segregation from the heartbeat, smell, sounds, because the baby even in utero can hear. In utero, there's visual information also beginning to come in, smell, sounds, etc. start factoring in very, very early. So that being the case, there's now been a turnover, and that actually came very strongly inspired by animal models, saying that there's way too much indication that survival rates will improve if you allow the infection risk is lower than the benefits that you get from. So in that risk-benefit analysis, people switch to actually letting there be much more physical contact between a premature baby and the mother or a caregiver, just to ensure improved survival outcomes. And there's a lot of clinical data that indicates improvement in those survival outcomes. So yeah, tactile information, so there's a lot of data on the tactile information.
General question. What happens to an individual who suffers from brain stroke?
Okay, so usually it depends on, so an area of the brain, either there is vascular leakage, so you have blood rushing into an area that causes damage, or there is a reduction in oxygen supply to that area. The brain is a structure that can't do without oxygen and can't do without glucose for any real substantial length of time. So it depends on the duration of deprivation from both oxygen and glucose and the degree of damage caused by blood flow into the area. I mean, so, yeah, so those areas of the brain that get damaged, depending on which area and what part of the body it may control, and the degree of damage, there is a possibility of recovery, or there is in timing, degree of damage, duration, the likelihood of getting in fast with like TPA, etc., the drugs that are life-saving, the gap being very short between the onset of the stroke and coming in with these drugs make a huge difference to the outcomes.
Well, I'm Dr. Shobha Nair. I'm a psychiatrist at BRC Hospital and not from the background that the rest of the audience is. And I just wanted to say that it was wonderful listening to you. And I felt that what we see clinically is what is being proven in the labs. So
you guys do the real hard work and heavy lifting. For us, we are removing the noise.
We have drifted a bit from being highly individual oriented and not looking at the psychosocial aspects. With a lot of pharmaceutical driven research, it's all become medications.
Yeah. I know there's been a strong push from the pharma industry to get their drugs out, though the body of data strongly indicates that cognitive behavior therapy is almost as effective as a medication for many, many conditions, and the combination is actually critical for recovery.
Absolutely.
Yeah.
So when we can look at everything as neurodegenerative diseases. Like Kreplin first said that schizophrenia was dementia precox, and then we went, no, it's a disorder. And now we have come back to say that it is actually a decline in cognition. And I like the way you answered about the resilience part. I was just thinking, but somebody asked you that question. Like all of us believe that some amount of early childhood adversities help us build resilience. And we feel that this is a generation which we are bringing up in the urban upper middle class, where we are mollycoddling the adolescents, and then they are all coming with different psychiatric symptoms with minimal stress. So what you say is that
it's very interesting you say this. They live in a much more complex world than many of us occupied when we were teenagers or children. There's this very interesting hypothesis by Robin Dunbar about how much your brain can cognitively handle socially in terms of the number of friends or close relationships you have. So we are a social species, so are rodents, and that initial circle is something like 5 to 10 people, close, intimate relationships where those people will drop everything for you at 2 o'clock at night and show up, right? That's your first circle. Then there's a circle outside of that, which is about 15 to 30 people, people you can still call but may not be your first middle of the night call. And then there's a circle of people outside that 150, 200 people you know well enough that you remember their name. Think about the world of our kids today. Their worlds have reduced on the first circle and expanded on the last circle to thousands of people on social media with zero intimacy in that first circle of 5 or 10. So this is a disproportionate social stress that they're going through. Our brains are not cognitively available to handle that large a social universe. We just don't have the bandwidth for it. So it actually becomes a social stress rather than a social support. And because we're outsourcing a lot of our life to our mobile phone, and the worlds that we occupy are going online and digital, it changes what you can socially manage. So it's a really interesting question that neuroscientists are now thinking about as to what is the strong social stress that comes from an online world that you occupy, where a lot of your social interactions are defined by that space.
And really appreciate your emphasis on socially disadvantaged and discrimination, which is very less spoken on any podium.
I mean, that I think is inspired by what I read about Shri VG Kulkarni. I was thinking, how is this relevant for a VG Kulkarni memorial lecture? And because of what I read about that focus on that, I wanted to, start the talk with that.
All right, I do see many questions, but I think we should continue the session of particularly responses in the tea break. So let's give her a break for a minute first of all. Okay. Yeah. And the last segment is vote of thanks. So I'll start with thanking Professor Vinita Vaidya for giving such an insightful talk. And it has actually given us perspective of seeing how our life experiences can, and particularly in the early years, can tweak and shape our stress response. And I think at HBCSE this is particularly relevant because many of us deal with school systems, teachers and educators. So I'm sure that we kind of realize now how our shared responsibilities are as a researcher and educators towards seeing what we can do in this aspect. Particularly, what I learned is it might be not just for social or community environment, but we also need to think a little bit about exosystems and also probably the macro systems in general, like when we are thinking as an educator. So I take again this opportunity to thank VGK's family and particularly Anita ma'am, who ensures that her support is there every year, and for the love and affection she has towards every member at HBCSE. Some HBCSE alumni and other dignitaries are also there in the audience today, so I would like to thank all of them. I also thank our Center Director, Professor Arnab Bhattacharya and Professor Savita Large for organizing the 23rd VGK Memorial this year. And last but not least, thanks to members of Dean's Office, Center Director's Office, Public Relation Office, administration. Thanks to Manoj for making the VGK Memorial poster and the invitations, also for live streaming and along with the help of other technical support team. And particularly thanks to canteen services, cosmetics, security, and all other staff, if I missed anyone, for the efforts to make this event really smooth and comfortable for all of us. So, and lastly, the audience. So let's again continue this discussion, but we will have it over tea. It's arranged just outside in the canteen area, the old canteen areas.